right, good evening, everybody. Thank you for coming tonight in the rain for this event, and welcome to the New York Genome Center. My name is Michael Zody. I'm the Scientific Director of Computational Biology here at the New York Genome Center, and I'll be serving as the moderator for tonight. I'm going to say a few words and then introduce both of our speakers, and then I'll hand it over to them. So I'm going to introduce our speakers in reverse order. Our second speaker tonight will be Dr. Evan Eichler. He's a professor of genome sciences at the University of Washington, an investigator of the Howard Hughes Medical Institute, and also an associate member of the New York Genome Center. He's also a member of the National Academy of Sciences. He has his PhD in molecular genetics from the Baylor College of Medicine, and he's a leader in the field of genome evolution and the study of structural variation, and particularly its relation to human disease. Our first speaker tonight is Dr. Wendy Chung. She's the Kennedy Family Professor of Pediatrics at Columbia University, and also the director of the Pediatric Neuromuscular Network Molecular Core, the New York Obesity Center Molecular Genetics Core, and the Precision Medicine leader at the Irving Institute. She's also the director of clinical research for Safari at the Simons Foundation and the principal investigator for the SPARK Autism Research Program. She has a PhD in genetics from Rockefeller University and her medical degree from Cornell University Medical College. And as you can tell from her many roles, she does research in a wide range of medical conditions, including obesity, diabetes, heart disease, and developmental disorders, including autism. Autism is an area of research that's very key to the mission of the New York Genome Center. We've sequenced over 20,000 whole genomes of autistic individuals and their families to try to understand the genetic causes of autism. And both Evan and Wendy are collaborators of ours, as well as world-recognized leaders in the research of the genetics of autism. So with that, I'd like to invite Wendy up to give our first talk. Thank you.
So I'll give a little bit of introduction. Probably many of you already know something about autism because it's a pretty common condition. So 1 in 59 Americans carries a diagnosis of autism. One of the challenges we've had, though, in researching it is that it's one sort of umbrella designation that's given to individuals with a wide range of both skills and talents as well as challenges. And so trying to research something that's so heterogeneous has proven challenging in terms of being able to replicate results, being able to have really robust findings, and ultimately to understand the biology to support individuals. It's challenging because to date there is no treatment that supports the core features of autism, so any of the social dimensions or difficulty in terms of social communication. So it's an area, in my opinion, of unmet medical need. With this, there are also many associated features. So there's some individuals, for instance, with autism who were unable to speak. They're nonverbal. They may have very low IQ. They may have epilepsy, even brain malformations or other challenges, health challenges. There are some individuals with autism that are, in fact, really quite brilliant, but still have challenges in terms of that social connectivity and communication, but do have really unique ways of the way they see the world, the way they interact with the world, and ultimately the way they solve problems. So it's a heterogeneous group, as I said, but one that I think is really worthy of being able to do what we can in terms of supporting the diversity of our community here. As I said, because of this, it's been challenging. It's also been challenging because when you think about this, what does a mouse with autism look like? It's been hard in terms of some of the behaviors to be able to encapsulate that and really have good animal models of this. And so ultimately, humans are really, really important for us to be able to understand this. And genetics has provided a framework for us to get a really strong anchors and foundations in terms of understanding the molecular biology and the genetics across this heterogeneity so we could make some progress in terms of supports. So as we did this, and I want to give credit where credit is due, folks like Evan were really instrumental in terms of a lot of the foundation for starting to think about especially one specific type of genetic change, that of de novo variants or new genetic differences. But as we started thinking about it and started thinking about, for instance, some of the foundational work that was done by the Simon Simplex collection, it was, at the time, I think, a really impressive cohort of individuals, very well characterized, very well genomically characterized ultimately, but still somewhat limited in sample size, so about 2,700 individuals, either trios or quads. And we knew what we could find with that number, but we knew it wasn't enough. So Evan and others had done calculations in terms of estimating the number of genes that were likely involved, and the numbers ranged somewhere between 5, 6, 7, 800 genes that might be involved. And to be able to really saturate at least the de novo space, we knew we needed to be in larger numbers, approaching 50,000 families, so trios or quads, and ultimately, as I'll get into, to really understand the genomic architecture well, requiring even more than that. Up to, my current estimates are about 200,000 will really be what we need to understand many of the inherited variants, both common and rare. So as we did this, one of the things that I love about being at the Simons Foundation is we can really think about what the scientific questions are and address them. So really being able to think about what the community needs and being able to come up with solutions. So that's the genetic dimension. I also want to be very clear, though, that beyond this genetics, everything, even though I'm a geneticist, everything is not in the genes. And to be able to understand some of these other modifying factors, the things that are important in development, both prenatal as well as early childhood development, again, there's heterogeneity in those etiologies, and one really needed sample sizes. In addition, when we started thinking about treatments and supports, clinical trials, interventions, you need an engaged community to really be able to pull this off. And so one of the things that was frustrating for me at the foundation was that we would give really great scientists grants to be able to do many of these, if you will, human subjects research, but research that involved individuals, being able to do imaging and EEGs and study natural history and interventions. And it would be just incredibly frustrating when they weren't able to actually complete their studies because they couldn't recruit. They couldn't get the right individuals that met their inclusion-exclusion criteria. So we had this bold idea, which was to create really a mega sort of, I think of this as the Framingham of autism, but being able to come up with ultimately the driver for autism research in humans for decades, I hope to come. So we called this SPARK, Simons Foundation Powering Autism Research for Knowledge. This is set up as, and you'll see some of the numbers in a bit, but meant to be a cohort in the United States that was engaged with us for the long term. So they are all recontactable. And in fact, in many cases, we've recontacted them many times. Incredibly well genomically characterized. So now one could start parsing this out and getting rid of some of the heterogeneity and get homogeneity to a certain extent. And again, being able to see this longitudinally. So individuals change, they have different challenges, and being able to see how this evolved over time. And so as I'll show you, we've set that up. And one plug I want to make very clear for any of you who are genomicists or researchers in the autism space, this is all of this data, all of the ability to contact this is freely available to the entire genomic community or entire scientific community. We want these individuals and these data to be used. As we think about this, there is a heterogeneity even in the genetics of autism, which is there are some things that are powerfully genetic, single genetic factors that have a very big influence, but there are also a lot of things that are little tiny bits of genetics. So in other words, common variants, which in totality really determine potentially a risk in terms of autism. Interestingly enough, towards the left, those that are highly penetrant, oftentimes de novo mutations, are skewed towards individuals who have more cognitive challenges, many of them either intellectually disabled or associated with a lower IQ. The polygenic risk, some of these common genetic factors, interestingly enough, associated with high academic achievement. In fact, just the opposite to a certain extent. And it's really, I would argue, the combination of those factors together, plus one's exposure over a lifetime that ultimately determine how the brain is functioning and how behavior is tied to that. As we thought about this, we thought about being able to characterize individuals with autism, but then once they got a diagnosis, once they got a genetically defined etiology, that was really just the beginning for them. That was the beginning of what we hoped would be a next phase for them, trying to both understand themselves, understand how this, what challenges might be ahead, but more importantly, what we could do to support them. And so we've also created sort of the next chapter in this, which we call Simon's Searchlight, based on what the families had told us. They saw themselves as a beacon of hope, being able to shine line in terms of their search going forward with answers and hopefully brighter tomorrows. As we did this, the eligibility criteria are very simple. Spark is meant to be very simple and accessible to anyone. So the eligibility criteria are easy. You have to have a professional diagnosis of autism. So that could be your neurologist, your pediatrician, your psychologist, psychiatrist, whoever it is, but giving you that diagnosis. And that's really, at this point, being able to do these online surveys, either with help or do it on your own, but do this from anywhere in the country. We have this set up with 31 clinical sites around the country because we believe these clinical sites, they know autism, and so we've invited them to recruit their patients at those sites. But as well, you could do this from anywhere, on your smartphone, on a tablet, be able to do this. At this point, and this is actually, these numbers are even a little bigger. At this point, we have over 185,000 individuals within Spark. We've been doing this for a little bit more than 3 years. Within this, we've now topped 70,000 individuals with autism. And we haven't got all of them as complete trios yet with the biospecimens, but we're moving forward in terms of doing that. And as we've done this, I'll get to it in a second, but we have this mechanism called Research Match, which is allowing participants in Spark to be able to be part of anyone's research. And again, anyone can come to Spark, being able to say, I'd like to have, these are my eligibility criteria now for my other study. Can you invite them to participate? And at this point, we've had over 40,000 families invited to such studies. As we do this, this was a new way for me at least of doing recruitment. We're extremely data-driven in terms of doing this. So we know every single week if we put out something on Facebook, if we put a particular visual, if we have a video of a certain family, we know how it tests. We can do A/B testing in terms of knowing exactly what works better than another, if our messaging is clear. I have to say it's been fun just being able to be that part of that data science. As we've done this, we've realized that we really do have largely, at least in the pediatric group, a representative cohort. We're sort of, the distribution is what we see generally in terms of individuals with autism. That is that individuals tend to be school age for the most part in the cohort. We have fewer adults. It's an area I would like to have more of, but we will get this over time, I'm convinced. And we have about 30% of the community that's intellectually disabled, again, what we see in terms of the general community. You can see on the left the exact distribution by IQ score. I think importantly, for any of you who are interested, we have several individuals who are actually very, quote unquote, high functioning, actually in the genius range in terms of IQ, as well as when we think about educational levels for our adults, many of ours that are, have attained high education in terms of college and beyond. In the interest of time, I won't go through too much detail, but we do have largely demographically also a reflection of the United States. So I think we've done a good job in terms of being able to be inclusive with this. And just very briefly, I'll say that as we've done this, it's been our goal to also give back to both individuals in our research as well as to the community at large. So we have ways of giving them back as they fill out surveys online, giving them back a report of how they did, where they are on the curve. We have also group reports to be able to say what we've learned from the community. And with this, we hope that this is something that continues to engage and retain our community because, again, we're with them for the long term. Just briefly, we have done pilot studies and now full-on studies in terms of understanding the genetics. Just to make clear what I may not have been clear enough about, as we find a genetic cause of an individual's autism, we actually give them that information back to them. We do it all in terms of within a sort of clinical standard. So we use a CLIA laboratory to do that. We use certified genetic counselors at our expense to be able to return and explain that information to people. We have very detailed, we call them gene brochures, but 5-6 pages on each condition to empower them with what does this mean, what do they do with their next step. On average, as we've been doing this in the pilot study, which has included exome sequencing and genotyping, and in some cases in the future, thanks to the New York Genome Center, we'll also include whole genome sequencing and part of the cohort. We're running about 10% of the cohort that has things that we are 100% sure are a major contributor to their autism. So we have a medical genetics community that vets and reviews every single case and releases it. You'll see from this in terms of the types of genetic findings that we have, about half of them at this point, or about a quarter of them rather, are at copy number variants, so relatively large chunks of DNA, whereas we have many more that are single gene differences, many of them de novo at this point, although again, as we understand better the genes, we'll also be, I'm sure, understanding the inherited variants as well. In the interest of time, I'll just say very quickly, within our first pilot study, which is now published, we did 457 cases or families. We identified many of the same genes that we were used to seeing, and it was very good validation for us that this method of ascertainment was very similar to other autism cohorts. Some had wondered if we had this just sort of volunteer army, was this going to be really reflective of what we see? And the answer, I think, is yes. There were concerns by collecting saliva. In fact, would we be able to get good, high-quality data? I would say, in fact, the answer was yes. We did get good, high-quality data. So for then any of you who want to use it, I hope you'll feel reassured by that. And we did start to identify on the right some new genes that we hadn't previously been certain about. As we did this, we have now the next wave of data, and again, this is an unabashed advertisement for anyone who would like to use the data. It's very broadly consented, actually, for any use, autism or anything else. And so now in this next tranche of data, we have about 6,400 families that have been analyzed. One of the things that was different than the Simon simplex collection is we have both simplex as well as multiplex families. They're distinguished, so you know how to think about that. This allows us to do some different types of analyses in terms of especially inherited variants. So we're looking forward to the community also being able to chew on that. And you'll see just the distribution of where individuals are around the country. So this is some analysis, and I think Evan's going to talk about some slightly different portions of the analysis. But this is looking at this sequence variants, so none of the copy number variants will I speak about. But again, in terms of seeing where the signal is, here we have the likely gene disrupting variants. And again, as we see, let me orient you to, these are all affected individuals with autism. In green are our simplex cases, in blue are our multiplex cases, and then we have several unaffected siblings as a comparator to be able to compare to. The main thing I want to point to is that there is a difference we see between the simplex and the multiplex cases, and perhaps this is not surprising, but we see a greater burden of de novo likely gene disrupting variants within those simplex cases. And you can imagine that one reason you might not have a family history of autism is because, in fact, the genetic event is brand new. And so, of course, if it's new, you're not going to see a family history of antecedent generations. On the other hand, though, I will point out that this is not zero. And again, comparing it to the unaffected siblings, in fact, there's an enrichment even within the multiplex cases for these de novo variants. And I can say, having seen and now talked with many of these families, there are, even within the multiplex cases, these new de novo genetic events, which are the same as we see in simplex families, but within the context of multiplex families as well. So again, just in the interest of time, I won't go into too much detail, but to say that as we've now done the TADA analysis and compared this on the right, these are the results of the SPARC analysis. On the left are large cohorts, including the Autism Sequencing Consortium, the Simon Simplex Collection, looking at the top 20 genes. The point that I want to make is these are very much almost a mirror image. So in other words, the findings that we've seen in other cohorts, ascertained other methods, very similar to what we're seeing in SPARC, likely gene damaging variants in red, missense variants in blue, and again, the mix approximately the same. Slight differences, but approximately the same in terms of what we've seen. Now looking at the TADA analysis, and again, I know this is complicated, but at the top here, and each of these is one of the genes and being able to see statistically the signal. Safari genes were Safari class 1 or 2 or group 1 or 2, so things we were fairly confident of. Other genes that we know about from developmental disorder studies, as well as previous TADA analysis. In other words, these genes on the left, very, very high confidence, and then within now the SPARC analysis, the posterior probability very, very high. But importantly, what I want to draw your attention to are some of these white spaces. So these places where we haven't had genes identified before. The statistical evidence isn't quite as high for understandable reasons, but it now is getting robust enough to identify many, many new genes. And again, as we start to double the sample size in SPARC, many that I'm sure we're going to get into the range that is statistically extremely significant. Again, enriched in terms of likely gene disrupting de novo variants, but also missense variants, and in fact, in some cases, recurrent missense variants as well. So I'm just going to wrap up very quickly to say that with this, the biology is similar to some of the biology we've seen before. So I think what we're seeing is more genes, less frequent in some cases, but similar biology, specifically RNA biology, RNA binding proteins specifically, a lot of transcription factors, a lot of genes that are related to development, Wnt signaling, other pathways that we see within development. Interestingly enough, and I always think about this from a therapeutic lens, many kinases and phosphatases that I do think are more druggable, so to speak, as well as many channels. And then histone modifiers, very fundamental just in terms of developmental biology. So I'm going to skip ahead just very quickly to say that knowing this information has been very helpful for us in terms of understanding prognosis. One of the things that's been really eye-opening to me only in the last year is to see that the natural history of these is not all static, that some of these are actually degenerative conditions. Some of these we now realize are some of the same genes we see in atypical Parkinson's disease, for instance, but we now know that if you ascertain them from young children instead of from individuals who are in their, say, 50s with PD, in fact, they're the same genes, and we can literally see within the brains some of the atrophy and in some cases the deaths associated with this. So it's helped us in terms of understanding biology, but also to understand what our sort of constraints are. Finally, I'll just round out by saying that, as I said, we have now these searchlight communities. These are the number of individuals we have in these searchlight communities, each of these genes specific, the families learning from each other, us learning from them and learning together. We have repositories in terms of cells, data, longitudinal data, ability to recontact. So if any of those happen to be your favorite genes, please let us know in terms of being able to do more with them. The final thing I'll say is just that we're also getting into treatment. So with this, with the 16p11.2 deletion, we are just getting ready to launch a clinical trial using R-baclofen in that specific group. We started out by using animal models to ensure that at least in an animal model, we looked like we had appropriate response. We did that with multiple animal models to show the robustness. And there are now studies actually going on both in Canada, EU, in Europe through EU AIMS, and now in the United States. And as I said, ours is specifically with one group. As we do this, it may be that for some of these conditions, early diagnosis is going to be critical for early treatments. And so I put this up here in part because we have some experience with early diagnosis from newborn screening for other disorders. We've done this for spinal muscular atrophy. Yesterday, we just launched for Duchenne's muscular dystrophy, and some of you may be reading about this in the press. And there really is the ability to do remarkable things when you know about this early on. If I can get this video to play. This was the first little girl that we diagnosed with type 1 SMA, or would have been type 1 SMA by newborn screening. She was able to get a medication, nusinersen, and would have died by her first birthday, but instead these are her first steps at 14 months of age. She's now 3 years of age, a thriving toddler, strong, smart, and so I do think there is the ability to change the natural history, especially for some of these degenerative conditions if we get in early enough. So as we do this, I hope what we're doing is, for those of you who don't really care so much about autism, that's okay. I would put this forward actually as a paradigm for what you could do in genomic medicine for many, many different diseases, both common and rare. We've thought a lot about this in terms of what it takes to scale this, what it costs in terms of doing this, and it is actually, it's expensive the first time you do it. I won't belittle that. It's much, much cheaper as you know what to do and what works and going forward. And so I'd encourage you all to think about this for your diseases if autism is not your disease. If autism is your disease, we've got it made. You don't have to replicate this and reinvent the wheel. Just come to us and take the data, take the resources. As you do this, I just want to say that it's been great, mainly working with the families in terms of doing this as well as the teams. I just want to put a shout out to some real important people who've been major parts of this in the audience today. Yufeng Shen, who's a colleague of mine at Columbia, Shui Ya, as well as many of the members at the Simons Foundation, and let me see if I can point her out, Pam Feliciano very specifically, and Amy Daniels over here, two of the ringleaders for the program, but a cast of, it feels like, thousands.
So I'm going to skip over some of this because it's all more or less been mentioned, but just to emphasize that autism, at least the way we think about it, is really an umbrella disorder that involves many comorbidities, neurodevelopmental delay, epilepsy, as well as neuropsychiatric features. The genetics of this disorder is well established, and it comes from identical twin studies, and it comes from essentially relative risk from SIBS, with the general population prevalence about 1%, or a little bit more than 1%, and you see that identical twins have a very high risk of actually developing essentially the disorder if one is diagnosed. The other feature, which I'm not going to talk much about, which is very characteristic of this disorder, is the male bias. As individuals get higher and higher functioning, that is to say in terms of their IQ, the actual male bias becomes more and more predominant. I would argue that probably the most significant finding in terms of setting us, at least geneticists, straight in terms of how to think about this disorder was early work in now 15 years ago, which is the discovery by several people such as Mike Wigler and Jonathan Sabat, as well as others working in the developmental field like our own lab, is that in fact children with autism and children with developmental delay have an excess of large de novo, typically de novo variants that are copy number differences. So in children with autism, there's about a twofold increased risk that they actually have a large, what we call a CNV. And this was important for a couple of reasons. One, it taught us that essentially de novo variation was going to be very important for thinking about this disorder. So these weren't necessarily inherited. They can be inherited, but many of them were de novo in the sense that they weren't seen in mom or dad, but they were actually in the child. And the second was that dosage imbalance. So having half amount of a gene or having a little too much of that gene are going to be important for thinking about the etiology or the molecular basis for this disorder. So when next generation sequencing technology came along, around 2010, it really took off. The ability to go after these families that we had previously screened largely just for large copy number changes really, I think, revolutionized the field. So this was an effort of many labs, but we were one of 3 that targeted what was called the Simon Simplex collection, 2,500 families, which involved individuals that did not have a family history of autism, where there was one affected, it's highlighted here in black, and there was data for about 2,000 of those families with both mom, dad, and an unaffected sibling. And this was really the design of Mike Wigler. Some of you know Mike. Mike's basically argued that we needed to have a genetic control for these experiments, and an unaffected sibling without autism, in his mind, and I think a lot of us are convinced by it, is one of the best controls that you could have within a family. So the experiment was very simple. Find the de novo mutations, the sporadic mutations, in the child with autism, shown here, and compare it to the unaffected SIB and ask if there was a difference. And so, not to bore you with this detail, but this is the upshot of several papers that were published, and it kind of culminated with this one with Ivan, Brian, and Stefan, which just basically classified the mutations, the de novo mutations, into 3 types. Those that were likely gene disruptive, so gene-killing mutations or loss of function. Those that were synonymous, i.e. mutations in a gene that had no difference in terms of the amino acid that was going to be produced, but it's still de novo. And those that are missense, so a change in essentially amino acid composition. And the really cool thing, and now highly reproducible result, was this, is that in children with autism, there was a 2-fold excess of gene-killing mutations that weren't in either of the parents compared to the unaffected SIB. It wasn't an overall increase in mutation rate, because you can see the synonymous burden or rate of mutation is the same for these de novo mutations. In missense, believe it or not, people did not believe it back then, but they now believe it now. There is an excess of missense, de novo missense mutations in children with autism. But when you look at them, they typically tend to be the more severe missense mutations, so the ones that were predicted to disrupt function. So that was the good news. 2-fold excess of de novo loss of function mutations in children with autism. The bad news, as Wendy already alluded to, is that we almost never saw lightning strike twice. So in that group of 2,500 kids, we only had a few genes where we actually saw different families have the same mutation. And that's actually really important, because we don't want to have these aggregate statistics. We want to be able to say to a family, this is the cause of autism in your child. And for that, we need to actually sequence and characterize not thousands, but tens of thousands of individuals. The other thing that came from this, which was actually, I think, really important, and I actually wasn't a believer in this when Brian O'Rourke, a postdoc in my lab, first started doing these experiments, was that when you look at the genes that are disrupted, even though they don't have power for the individual gene, if you ask a question, how do they work with one another? So what I'm showing you here is protein-protein interaction, where the actual size of the circle represents the number of individuals in that cohort of 2,500 that either had a loss of function or likely gene disruptive in red or a severe missense mutation in blue. What you found was that the genes were not randomly distributed in terms of where, in terms of their function, but they actually focused or converged on specific functions. So this is showing you what we think are some of the key functions that are the genes that we're identifying. Genes important in terms of the functioning of the synapse, long-term potentiation. Genes important in terms of Wnt signaling, cell growth, and genes important in terms of chromatin remodeling. If you went back to that original study and you projected all the mutations that were loss of function in de novo, 65% of them in those families that we identified are a step removed from this kind of core network of genes involved in chromatin remodeling, synaptic plasticity, and Wnt signaling. So, getting at this question of trying to find the individual genes, we took two approaches. One approach was to sequence tens of thousands of individuals, targeting specific genes that we identified as having one de novo occurrence to find additional examples. The other approach is just to do more exomes, which is the approach that Wendy basically outlined. And at the beginning of this year, we had basically took an approach, we actually did both approaches, but we decided to actually combine data from both the autism cohorts that had been sequenced and published, as well as children that had developmental disability, and treat them as one cohort, neurodevelopmental disability. And when we did this, we identified about 253 genes, of which 124 met statistical significance by the most stringent of criteria. So they would keep the Broad people even happy that we had in fact established significance. And this is just kind of a listing of all those genes, with the lower numbers being essentially less significant as you move down. Relatively few missense genes at this point, but a large number, relatively large number, if we estimated 400 or 500 de novo for loss of function genes associated with loss of function mutations. So what's the point of this? Well, the point of it is that once you prove significance of a given locus or given gene, you can now go back and actually justifiably recontact and reassess the families that have a specific lesion. So these are some new examples of genes that we've been characterizing over the last year. This is a gene called TANC2. It's a tetratricopeptide ankyrin repeat coiled-coil containing gene. It means almost nothing to me in terms of its description, but we are convinced that it's actually relevant to autism and developmental delay. And so here are the data. We've identified now it's close to 20 families that have loss of function mutations. Slightly more than half of them are de novo, so they're not inherited, but they're essentially de novo mutations compared to essentially the general population, where 45,000 individuals in the population have been screened, only one loss of function mutation has been identified. So we consider this to be a likely high impact or a highly penetrant gene. The phenotype of these individuals is interesting. They're on the more severe side. They have often delay in language, they have autism, variable levels of intellectual disability, but they also have motor developmental issues as well as predisposition to seizures, which is actually quite common in this group. A subset of these show craniofacial abnormalities, but not all of them, which is actually, this is fairly common. Some of the clinicians have referred to this as a Rett-like syndrome. So this one is particularly interesting because the biology of it actually fits fairly nicely with what we think about in terms of being a synaptic function. So TANC2 is a scaffolding protein that interacts with the PSD95 complex on the dendritic spines. And the data that's fairly recent suggests that it's important in terms of vesicle transport, specifically at specific sites in neurons at the locations of synaptic neurotransmitter release. It's highly expressed in newborn excitatory neurons, but it's expressed broadly in the brain, but very highly in those specific ones. Here's another example. This is it should be a new gene to many of you, CSDE1. It stands for a cold shock domain gene, E1 subunit. We have found an excess of de novo and loss of function mutations in this particular case. Once again, showing here are the statistics at the bottom. Interestingly, again, about half the families are de novo, but another half of the families we actually see transmission of these, essentially going from a parent to essentially the child. In a subset of those families, we see that the parent that's actually transmitting also has neuropsychiatric features, often fairly severe, including diagnoses of schizophrenia, major depression, but they're not consistent, and they're certainly not autism, at least clinically defined. So there seems to be some relationship for these genes that are maybe 50 to 70% de novo mutations, but also showing transmission, and parents actually showing some features, but not necessarily neuro development in the traditional sense of developmental delay or autism. So we've done some experiments, actually using some of the tricks of the trade developed here by, or at least in Rockefeller by Bob Darnell, looking at what this gene actually does. The gene produces a protein. Hits-Clips assays actually shows that, in fact, it's an RNA binding protein. And in fact, when you look at the genes that it, in fact, or at least it binds with at the RNA level, there's significant overlap with other important genes important in terms of neurodevelopment, such as FMRP. So there's a highly significant overlap in the genes that are being recognized by FMRP and CSD1. Then you can do experiments like knock this gene out. So we've done it in zebrafish, we've done it in actually mouse cortical neurons, and this is showing you experiments actually of knocking this gene out in Drosophila. And what you're supposed to get from the right here is that one of the things that we see when the gene is knocked out, both in the heterozygous condition as well as the homozygous knockout, is an increase in the number of boutons at the end of the neurons. So you can see these spines, and then here indicating is essentially this excess of boutons. Boutons is where the synapses will form. So there's too many synapses that are being produced. The other feature, which is quite interesting, is that if you look at the actual morphology of the neurons in the fly that are knocked out, you'll see that their axons are significantly longer in both the knockout as well as well, this is just showing two different knockouts of CSD1, and it can be partially rescued by the addition of the wild-type gene back in. This is kind of classic behavior for immature neuron development. We see this with FMR1, and we've seen it with many other RNA binding proteins. They're important for maturation of neurons during development, and if they're not present or not there enough, they basically result in essentially this type of phenotype, kind of immaturity, too many boutons, too many dendrites, too many spines, too many or too long of axons. So this is part of the kind of the challenge that Wendy already mentioned, is that as we are sequencing now additional exomes and actually families, we have discovering, at least the hope is to discover more genes under this kind of model of de novo mutation, because we simply have to sample more and more individuals in order to actually prove a given gene is significant. We did some modeling based on combining both ASD and developmental delay cases of 10,000, and our data suggested that even when we get up to about 25 and 30,000, in terms of the red line here, which is the loss of function de novo mutations, we're going to start to asymptotically kind of reach a maximum. So we're going to start to kind of max out on the types of genes that are going to have de novo loss of function excess. So that will be the lowest hanging fruit, but it's still pretty high up in order for us to access. And as a comparison, we just analyzed and combined the 10,000 cases that we had with the 6,499. We're working with Wendy on the SPARC collection. And it identified about 94 additional genes at a 5% false discovery, and 45 of those genes would meet exome-wide significant. So this is a lot of sequencing to pick up 45 genes. Very important to pick those ones up. New genes in there, new biology to be discovered. But you can tell that by the curve here that this is going to plateau out. So I want to kind of end with essentially what I think is an important next step. It's something I've been thinking about for at least 5 years, of not just de novo mutations, but actually thinking about a different class of mutations. We call them in our lab private mutations. So these are inherited mutations that are being transmitted within a family, but they have a very kind of specific feature, is that they are found exclusively in that family. So this would be ultra rare. Well, the most rare is de novo, but these are the next step. So the way to think about this is essentially mutations that have originated maybe in the grandfather's generation or generations before. And now when you compare against all your autism families, you only see that in that particular family. So we call those private mutations. The reason I'm interested in these is based on an observation that one of my students made a few years back when we were doing the Simons collection. He went back and everybody was focused on the de novo mutations, and I said to him, why don't you look at the inherited mutations? And he said, well, that's a fool's errand because we won't have power. But I said, look at the different classes of mutation, loss of function shown here in red and the gray being the missense mutation. And I want you to break down the mutations based on their minor allele frequency, based on the autism population of 2,000 families that we're characterizing. So he did. He broke them down by 0.2, 0.15% allele frequency, all the way down to the private, which are the ones that are restricted to a given family. This is what he saw when he compared the affected versus the unaffected. So remember, this is the Wigler design of having both of those cases, both the unaffected and the affected, asking a simple question: is there an excess based on a minor allele frequency of different classes of mutation? Nothing for less than 1%, nothing, no difference between proband and siblings for less than 0.1%. But if you went to private mutations, you saw this signal, and it was significant. And what made me a believer on this is that if we went back and looked at the genes based on their tolerance, and we used a score called RVIS developed by David Goldstein back then, we saw that the genes that, as you went from tolerant to essentially least tolerant, so that means they don't allow for mutations in the general population, this odds ratio went up, specific for loss of function. So we wondered, and the new genomes that are being sequenced by the New York Genome Center, the CCDG, and also with Wendy's Spark collection of 6,500 families, do we see any evidence? Can we basically replicate this initial finding? Now keep in mind that this CCDG set includes all the original SSC that we studied in that very first paper in 2015. So this is, for those of you interested in the numbers, this is the number of private mutations, this column here, this LGD, the average number we're seeing, and it's really important to consider your population background because you're only as good as your depth in terms of the number of individuals. And so the average number of loss of function mutations in a South Asian, interestingly, is higher than even in African, but lowest for the Europeans because 89 or 85% of the samples that we have are European descent. So definition of private is dependent upon the size of the cohort with related ancestry. So that's an important consideration. So in other words, these mutations would generally be older, many of them, because we just don't have enough South Asians that have been surveyed. So we've kept that in mind as we've done these analyses. But let me just share with you some of the results. So we went back and we looked at the CCD genomes, looking at 4,362 autism versus 2,235 siblings. And I think you can clearly see that this is now using essentially Daniel MacArthur's definition of loss of function intolerance, so PLI. And you can see that as PLI actually increases, you can see that this odds ratio is beautifully increasing, suggesting that there's an inherited signal of private mutations. Private mutations are more likely to be found in probands than the siblings. So each of these comparisons is the probands versus siblings. This is Wendy's Spark collection, and you can see basically a mirror image of these results. You can see this basically increase in the odds ratio. So the observation has been replicated. People actually criticized us back then and said that can't possibly be true. We're not underpowered, but in the aggregate, you have power. These are actually now very significant findings in terms of the transmission of private variants, preferentially to children with autism. So we asked a separate question. We said, well, we already know the genes, at least a subset of the genes for de novo burden. However, if we look just at those genes that we know are associated with de novo mutations in children with autism, do we see any increase in terms of proband transmission or transmission to probands compared to siblings? And the answer is yes, we see a difference. So we've compared different sets here. We have our own very conservative, the most conservative set we have is this 124. We have this set that we got from the Spark group. This is the 249. And you can see that the odds ratios increase, at least in terms of numbers, but many of these don't quite reach significance because the numbers aren't quite high enough yet, at least for those subset of genes that we're looking at. So that's the result for looking at the genomes. This is the result, once again, looking at the actual data from the Spark collection. You can see the Simon's Foundation, this set of genes, is not doing too terribly good, or well, at least by this analysis. So the Safari 845, which is a subset of autism risk genes, these ones are all established by de novo significance. They do not actually show a really dramatic increase in terms of the odds ratio for private variants. So we then asked a very simple question after that. If we remove all the genes that we know are associated with de novo mutations, what happens to our odds ratio in terms of these private loss of function mutations? And I was surprised by this finding. So this is again showing genes organized, and this is all the genes from the least constrained genes all the way to the most constrained genes. This is considering all genes, including the de novo, the ones that show an excess of de novo mutations in autism. This is now comparing the remaining genes. You can see that there is a little bit of a bump, but not much. The odds ratio still stays pretty high. In other words, most of the genes, we think about 85% of the risk that's out there that's from private loss-of-function mutations has nothing to do with the genes that we've already identified in terms of de novo mutation, which means there are a lot of genes out there yet to discover that are transmitted, but they have originated in a few generations ago in the family's pedigree. And then this is just to show you all those, basically excluding the known autism genes from de novo. I think we also excluded all the Simon's Foundation genes from this. And this is just comparing probands, the actual percentages that actually have basically a loss of function mutation. And these are the actual numbers between probands and siblings as we march down. So you can see the odds ratios are still appreciable. They're still significant. And then here's the last piece. If we ask the question, let's look at only those that actually have 2 or more loss of function mutations. Remember, the average European has 5 loss of function mutations that are private. The result is, the results become actually more significant and the odds ratios become larger. So I've been arguing this for a while, that it's not just, there's a space between polygenic risk and de novo highly penetrant, and it's oligogenic, where it's a couple genes, 2 or 3 genes that are probably contributing to high impact in these cases. And I think this result, we've now seen it 3 or 4 times with CNVs, and we've now seen it again with this, really argues for an oligogenic model of a few major hitters in a given individual, as opposed to thinking about these extremes of polygenic risk versus essentially de novo mutation. Last point I'll make, and I'm really excited by this, although it's at the edge of significance at this point. So I said to the postdoc that was working on this, I said, take all the genes that have a private loss of function mutation, remove all the known genes from Safari and the de novo list that we have, and take those genes that are actually seen transmitted 1 or more times to probands, but never seen in an unaffected sibling. So she did this, and she got this protein-protein interaction network, which for a lot of you probably looks like just a bunch of squiggles and lines here. But the interesting thing here is that in terms of the gene ontology, it's not random. We're seeing significance particularly for receptor tyrosine kinases and genes associated with exon guidance. And in particular, this particular pathway right here, this is a highly networked set of ubiquitin E3 ligases. And for those of you who know a little bit about the autism field, this is a set of genes that's very strongly implicated in terms of autism, particularly in terms of the proteasome pathway of degradation. So it marks proteins for degradation early in development. So we did this for Spark, and we combined it with Spark data, and then just because you can't see anything from this, just to show you that the results are getting more significant, and here's that network once again for the ubiquitin E3 ligase, protein ligase network. Almost each one of those highly interacted proteins there that you see is an E3 ligase. I don't think this is by chance. I think we're actually finding the inherited component, and the inherited component is pointing us to related but distinct pathways than what we've seen from the de novo. So in summary, I've talked to you a little bit about the large CNVs, kind of how we got into this field. We think it explains about 7 to 8% of cases with autism. For kids with developmental delay, that number rises to about 15%. Sequencing of protein coding regions, if we say that we believe there's about 500 de novo genes that have a de novo burden of excess, we've conquered or identified about 300 of those 500 at this point. Those last 200 will take all 50,000 plus that'll come from the Spark, probably to get access to. This genotype first approach, I've been a believer of this from the beginning, but the idea that you basically classify patients based on their molecular etiology, you then work out the detailed phenotypes after the fact, because I believe treatments are going to be geared toward the subtypes, maybe not at the gene level, but probably at the network level. And then this, I think this is the future, a focus on the private gene disruptive point mutations, especially in the conserved genes. Our data suggests that it's a different subset. There is overlap with the de novo, so clearly they can be transmitted in families, but there's a different set of genes and a different set of networks that are actually related to these private mutations. So I always show this kind of a scorecard of where we were and where we are, at least in terms of thinking about the genetic risk of autism. And obviously, we have a big green section here, which still remains the unknown. I would like to argue that the inherited private mutations are probably going to add another chunk of about 10% at minimum that we'll be able to understand by actually working these through. That still leaves lots of room for other types of genetic etiology to explain. But I think with the numbers, I think the locus heterogeneity problem will be solved. I think the recontact issues are really important, and I think proving those individual genes in a private or even a digenic model where you have multiple genes is going to be more challenging than essentially even a polygenic risk analysis. All right, so these are all the folks that contributed to some of the work I've done here. Most of the people in the center are the clinicians with whom we've worked for decades. The work I presented today from my lab came largely from Tin Yuen Wang, Wei Go, and Amy Wilford from my group. I also want to acknowledge Pam and Wendy for their excellent stewardship of the Spark, and then Tom and Mike for working together for the last, feels like a decade, but it's only been really 4 or 5 years, I guess, on the actual genomes. There's a lot more that we're going to gain from the genomes, and I didn't present all the CNV work on those, but I think if you really want to think about, if you really believe in a multi-model, a multi-mutation model of autism, you can't be satisfied with just sequencing the exomes. You got to move toward full genomes. All right, thank you guys.